Hi, now we want to discuss about DNA replication and PCR. So DNA replication is making DNA from a DNA template. So why do we need this? So when the cells divide from one cell to another, so one become two, we need an exact copy of its own genetic material, DNA. So that's when DNA replication is necessary. So think about this double-stranded DNA, and we unzip those, and how we can replicate those. So far, what we know is, and look at this side when we unzip. So this looks like a fork. So that's where we call it a replication fork. And we need an enzyme for producing this. So remember, DNA only makes from 5' to 3' end. So one side, one strand is 5' to 3', then the opposite side should be 3' to 5'. And our creation, making the DNA based on this template, it should start with 5' end to going towards 3' end. So this part is called a leading strand because this polymerization can happen continuously. So what does this job is the enzyme called DNA making it polymer. So we call it DNA polymerase. This catalyzes the reaction that adds a new nucleotide to the growing strand of the DNA, in this case, this. Then thinking about this problem, the opposite side has an issue because look at this DNA, the opposite side is 5' to 3'. Then to make a new copy from 5' towards 3' will be the opposite. While this fork has been elongated, we have only this continuously make it. So for example, like this is 3' end towards 5', then we should be able to have 5' to 3'. And then once this elongates, we need another exposed one. So we have to, again, make from 5' to 3' end. So likewise, when this will unzip, we have to have make it 5' to 3' DNA polymer. But this has to be discrete. So this is discovered by Okazaki, and we call it as Okazaki fragment. And while this is called leading strand, the other side is called a lagging strand. So DNA replication is initiated at sites called replication fork, which is called this look like fork structure. And DNA pore or polymerase requires here a short segment of nucleotide to initiate polymerization. So that we call it as a primer. And this DNA synthesis can only occur in the 5' to 3' direction. The lagging, falling behind, meaning this part strand can only be replicated discontinuously to form Okazaki fragments. So which much then then you have to join this together. So that tie-up is called ligate. So to form a continuous strand so we need an enzyme doing this ligation. So that's called, of course, DNA ligase. So this replication, and there's a number of enzymes involved. You don't have to remember all the enzymes, but pretty basic ones, I want you to understand those. So the double helix is unzipped and unwound. And then each separated strand. Let me show you here. So these blue ones are template, unzipped and unwound. And each separated strand acts as a template. So there's two templates for replicating a new partner strand, which is shown in green here. And look at this direction. So this is a leading strand, so 5' to 3' continuously, while the other side, you have to do it discontinuously, this part and this part. And then while doing this, nucleotide, so these bases are matched A, T, and GC. to synthesize new partner strands into 2 new double helices. So on the other side, look at this structure. This is a leading strand because you can start a primer here, a red one, and 5' towards 3' continuously, while the opposite side, called lagging strand, you can do it during this time discontinuously. So 5' to 3', 5' to 3', and you have to ligate those. So the enzyme machineries, there are a number of enzymes to do this. First, you have to unwound these helix. That's done by topoisomerase, and you have to unzip these DNA double strand. So that's done by helicase. And this part, if you look at closely, so 5' to 3', so the DNA synthesis can start from 5' towards 3' and so continuously. So that this part is leading strand. On the other hand, 3' to 5', then you have to have the opposite side. From here, a short part of primer, you kind of make this DNA and then DNA as Okazaki fragments, and to rejoin them, you need DNA ligase. So let's briefly overview the PCR polymerase chain reaction that has such an important role. So when we have a tiny pieces of DNA, which we want to confirm, let's say infection from coronavirus or something else, a disease, then we have to, replicate this to amplify enough amount of DNA so that we can test them correctly. So here is the original piece of DNA, original DNA, which we want to replicate. Then this is connected and we have to denature it first. For that, we need to heat that so that this DNA hydrogen bonding can be detached. And while we have this DNA primer and nucleotides around. So first, what we have to do is anneal at proper temperature, anneal the DNA primer to its proper position by hybridization. And this is the starting point for replication so that we can elongate at 72 degrees with these DNA nucleotide and polymerase. So the polymerase chain reaction happens, and on the opposite side also polymerase chain reaction. So one DNA strand become 2, and we repeat this one. So it's 1, 2, 4, 8, 16. So it keeps multiplying until millions of copies can be made. So this is a very nice, cool idea, and which led to Nobel Prize in 1993. So a good example of this is nowadays, okay, if some patient or some people is suspected to get infected by coronavirus 19, how to confirm that? And in fact, this PCR helps. But coronavirus is an RNA virus, while this PCR is for amplifying DNA. So you need to make this RNA the corresponding DNA first. That requires not transcription, but the reverse. So we call it as a reverse transcription. So to check that, we need a reverse transcription PCR, because making that RNA information of coronavirus into a DNA, and then we run this PCR, and there's called a real-time PCR, which we can see in real time during these cycles to check if there is suspected DNA piece or segment is present or not. So another slide, this is for reference for PCR. A technique to produce many copies of specific regions of DNA for further analysis. So let's go back here. Double-strand DNA, 5' to 3' anti-parallel fashion, and we denature it at 95 degrees, and then we anneal or attach primers, 2 primers at its complementary site, and then we extend by making this DNA polymerase working to make a copy of this. The important thing to recognize here is this DNA polymerization happens at 72 degrees, which is way higher than our physiological temperature. What it means is most of our enzymes are supposed to work best at our physiological temperature, such as 37.5 degrees, but this has been the key to find. And in fact, there are in a very hot place, there's still life forms around. We found the enzymes working at this temperature, that's called Taq polymerase. And because of this finding and use, we can actually run this DNA polymerization, and you keep this cycle over and over, and we increase the copy almost exponentially. So the components for PCR, you have to have DNA template and 2 primers, 1 and 2, which is primer means short nucleotide sequences that are complementary to the ends of the target DNA. So design of primers are important, and we do need DNA polymerases, especially Taq polymerase. And using this, we can amplify or even quantify the amount of DNA because we know how many cycles we went through, and this can be used for diagnosis of disease or microorganism, bacteria or viruses. Okay, next time we will get into more into detail of central dogma and how people have discovered the genetic code.